Imagine a world where we could study the intricate workings of our body's blood factory without relying on animal testing. This groundbreaking possibility is now within reach, thanks to a revolutionary human cell bone marrow model. But here's where it gets even more exciting. Researchers have successfully recreated the complex cellular environment of bone marrow using only human cells, a feat never achieved before. This innovation not only promises to reduce animal experiments, but also opens up new avenues for blood cancer research, drug testing, and personalized therapies. Our bone marrow, often unnoticed until something goes wrong, is a bustling hub of activity. It's where blood cells are produced, a process that becomes critically important when diseases like blood cancers disrupt it. Traditionally, studying bone marrow has relied heavily on animal models and oversimplified lab cultures. However, a team from the University of Basel and University Hospital Basel has developed a game-changing model that mirrors the human bone marrow's complexity entirely from human cells. But here's the controversial part. Could this model eventually make animal testing obsolete in certain areas of research? The bone marrow is far from uniform. It's a mosaic of specialized microenvironments called niches. One such niche, the endosteal niche, is crucial for blood formation and plays a significant role in blood cancer's resistance to treatment. This niche, located near the bone surface, includes blood vessels, bone cells, nerves, and immune cells. Until now, no human bone marrow model has captured all these elements. The research team, led by Professor Ivan Martin and Dr. Andres Garcia, has not only created such a model but has also published their findings in Cell Stem Cell. And this is the part most people miss. The model's foundation is an artificial bone structure made from hydroxyapatite, a natural component of bones and teeth, combined with human cells reprogrammed into pluripotent stem cells. These stem cells, guided by specific signals, differentiate into various bone marrow cell types in a controlled and reproducible manner. The resulting three-dimensional construct, measuring 8 mm in diameter and 4 mm in thickness, closely resembles the human endosteal niche. This model has sustained human blood formation in the lab for weeks, a significant advancement. But here's a thought-provoking question. As we move closer to human-based models, how will this shift our ethical and scientific approach to research? Professor Ivan Martin highlights that while mouse studies have been invaluable, this new model brings us closer to understanding human biology. It could complement or even replace many animal experiments in studying blood formation, both in health and disease. This aligns with the growing trend in science to replace, reduce, and refine animal testing. However, not everyone agrees on the pace of this transition. Some argue that animal models still have irreplaceable roles, especially in complex disease studies. What's your take on this? Looking ahead, the model could revolutionize drug development, though its current size may need to be miniaturized for high-throughput testing. In the long term, personalized treatments for blood cancers could become a reality, with individual bone marrow models generated from patient cells to test therapies. But this raises another question. How soon can we expect these personalized treatments to become mainstream, and what challenges still lie ahead? As we celebrate this scientific breakthrough, it's clear that the journey toward ethical and effective research is just beginning. What do you think? Are we on the cusp of a new era in medical research, or are there still hurdles we need to address? Share your thoughts in the comments below.